We're solidly taking an hour. Okay. No. Okay. Well, I think we will get started. Okay. Cool. All right. Well, welcome guys to the CNM seminar for May. So we're doing hybrid today. So we've got a nice collection of people in the room, and then I think we've got like another 22 or so online. So thanks for joining everybody. So I'm excited to bring to you today or this month some work that my lab has been working on. So this was a project that took a year or two to kind of come to ideation and come to fruition, but we officially started it this past September. So a lot of what we've done in my group has been working on nutrition models and metabolism models, but for the most part, usually for agriculture, the three main agriculture species, like, poultry, pigs, and ruminants of some sort. But I had, one day a great conversation with Scott Ciesler in the hall, and we said, like, hey, like, we should really do this for the equine sector. So we came up with this idea, and here we are today. So I'm going to present to you guys kind of an update on this project, what we want to accomplish, and where we're at. And I have two of my postdocs, Dr. Emily Leishman and Dr. Kara Cargo-Froome, who are working on the project right now, and they're going to kind of present kind of where they are at the moment with their work on the project as well too. So yeah, why do we want to build a model for horses? Maybe we should start there. Certainly models like this, models really become kind of where we, synthesize and analyze our cumulative biological knowledge, right? So, in any fields, we kind of do like feed them and weigh them, or, feed them and weigh them trials for, a number of years or decades. And, we realize that biological responses are complex. And so models help us to synthesize that and understand how the whole system works. They tend to, because of that, increase our biological understanding. So they can also be used in teaching settings. And they also help us to identify, research and knowledge gaps. So, as soon as you try to put math to something, you realize if you understand it or you don't understand it, or if you have enough data to build that relationship or not. And also, there's this nice interplay between experimental research and modeling where, both models help build better experiments, and experiments help to fuel and build, parameterize our models. And out of this, many, new research ideas can come. From a commercial, so these are probably on the top, probably all the, benefits to building a model from the academic perspective, but there's also big benefits from the industry perspective as well. Primarily, helping to us improve diet formulation. So in many of our big species, models are the foundation of our feed formulation softwares. We want to, reduce the environmental footprint of the equine industry as well. So some survey data that, for example, Mad Barn pulled together showed, that we're significantly overfeeding protein and energy in the horse sector. So is this kind of low-hanging fruit that we can actually move this sector more towards the idea of precision feeding and reduce some of this overfeeding of nutrients via better models. Improving equine performance and welfare. So, can we help reduce obesity, for example, and the associated health effects? And really, in the end, what we also want to do is make Canada a leader in, equine nutrition solutions and digital tools. So this is kind of like our broad picture objective of why we wanted to do this for the horse sector. So this project that we're working on, it's a big project, and we've kind of divided it into a few different modules, but really our overarching goal is to develop an equine digestion and metabolism models for horses. And to build this, we want to capitalize on the models that have been developed in other species. So we've been developing these kind of models for decades. And we can certainly stand on the shoulders of those giant models to say, to kind of spring forward models for the equine group. We're going to, use models from other species as starting point, utilize the published equine research to build and parameterize our models, and then where we see gaps, target experimental research to fill those knowledge gaps. So this is kind of the modular development. This is what we foresee needing to develop. Um, at the kind of the core is probably item 2 and 3, which is developing an equine digestion kinetic model and an equine metabolism model. Now, if we're going to develop a model to look at equine digestion kinetics, so how is feed degraded and how is that balanced against things like passage rate, characteristics of the feed, we need to characterize those feedstuffs. So one of my postdocs, Kara, is actually even this week working on a pilot to do some in vitro feedstuff characterization. So looking at degradability rates for protein, starch, lipid in the stomach and small intestine, and then looking at fermentation of those nutrient components within an in vitro fermentation system to simulate the hindgut. So Kara's going to talk a little bit later about some of her in vitro characterization or feedstuff characterization work. So that will kind of fuel a digestion kinetic model. And at the moment, my other postdoc, Emily Leishman, is working on developing the equine metabolism model piece of it. So, these three are kind of the big pillars that we're working on right now: feedstuff characterization, the equine digestion model, and the metabolism model. But certainly other things that will play into this as time goes on is looking at things like gestation and lactation. So how do we quantify the requirements and demands from the animal for these phases of life? And exercise and meal patterns. So looking more towards the high performance athlete animal. If we can model within day kinetics of nutrient delivery and nutrient demand because of performance bouts, can we assist target feeding or precision feeding those high performance athletes down the road? The piece that can kind of tie it all together is tying this kind of model together with an optimization routine. So we can certainly say, like, if this scenario, what do we predict? But can we loop that essentially to run, multiple simulations to say, okay, we want to optimize X, Y, or Z outcome. What should we feed, right? So if we can tie this model to an optimization routine, then we can ask the model to provide us back with optimized outcomes or suggestions for feeding. Tied into all of this, the one at the bottom is a component on KTT and GRIP, and I'll show you at the very, very end of today some of the videos, courtesy of Mad Barn, that we've been putting together to kind of talk about this project as well. So in terms of deliverables, what we foresee for this project, in terms of giving something to the equine sector, we want to deliver a digital decision-making tool, right, to help with things like ration balancing, performance optimization, minimizing environmental footprint, and also reducing the risk of disease. And we also want to provide this digital tool for researchers. So again, to have a place where we can contain our cumulative biological knowledge as a tool to explore hypotheses, identify research gaps and knowledge gaps, make Canada a leader in digital equine research. And we want to also generate the next generation of HQP for the sector. So one of the challenges that we've had thus far is that this intersection of equine expertise with modeling expertise really hasn't existed. So we have largely been trying to build that HQP niche to support this project. So we've really pulled together a really great team with diverse expertise to do this because we need expertise from so many different angles, and we don't per se have one person that has all of these expertise. So we've brought in cross-species modeling and animal systems modeling expertise, equine nutrition and physiology experts, in vitro feedstuff characterization experts, as well as expertise on the computer science and programming side. And then we've been doing this in really tight collaboration with our industry partners to ensure that in the end, what we're going to build is useful, right? We want to make sure that, if the industry needs an apple, we don't build an orange, right? They're both fruit and they're kind of the same, but not really in terms of what we want from them. So that's been really important. We've gotten numerous funding sources for this so far. So certainly it started with Mad Barn, but we've got funding support from LRIC, MITAX, and OMAFRA, and we're going to put in some more NSERC grants as well. And the other thing to just point out is this is probably going to be a multi-year, multi-phase project. What we're doing right now is kind of phase one in terms of building the base model. And we can from there see, what kind of problems are we coming up with, what knowledge gaps do we have that would fuel experimental research, and of course the next steps of the model going forward. So this is the roadmap, and what you're going to hear about today is from two pieces of this. So they'll probably seem a bit separate, but I wanted to give you kind of the big picture so you can see how these things all get tied together. But actually, Emily is going to talk to you first about the equine metabolism model and where she's at with that. And then Kara's going to come in and talk about feedstuff characterization and the work that she's been doing in the lab. And of course, the next leg that we're looking to start is the equine digestion model, which we hopefully will start work on this fall. So I think with that, I'll hand it over to you, Emily, if that sounds good.
Awesome. If everyone can hear me, that would be good. No, thanks, Jen. I'm excited to talk to you guys today about what I've been working on, I think for about a year now. And as Jen kind of alluded to, I am not an equine person, not historically. So I actually did my PhD in poultry physiology and not equine modeling. But actually, sort of what I've learned along this process is that sort of cross-species experience actually plays really well into working through these kind of problems. Because like Jen mentioned, there's not a lot done in the equine sector to date. So we really have to stand on the shoulders of models developed in other species and figure out how to apply them to the problem that we're working on. So it's been a fun ride, and I'm going to give you a little bit of a tour of what I've been working on sort of for the past year or so. But as Jen mentioned, there's in general across the board a lack of mathematical models for equines, certainly compared to the other main agricultural species, especially ruminants, swine, even poultry. And modeling at the whole animal level has largely been limited to 2 or 3 sources, mainly the NRC, as well as work done by Dr. Thomas Tulutke in 2011. And I'll get into those a little bit and just explain what they are. So a lot of you are probably familiar with the NRC, so the nutrient requirements, in this case of horses. If not in equines, you're familiar with them in other species. And this provides you essentially a lot of empirical equations to predict nutrient requirements of a particular animal at a single point in time. So you put in your information and you get your estimate of nutrient requirements for one single static point in time. So we would call this a static model. And it's also an empirical model because all of these equations, like the one on the slide there as an example, are really built from usually a regression analysis from experimental data. So you can see here these equations on the slide estimating the amount of dietary energy required by a particular animal at one point in time. You have a coefficient multiplied by body weight. And this coefficient adjusts depending on whether the animal is a low maintenance, an average maintenance, or high maintenance animal. So this works really well, but there's some limitations to models like this because they are based on essentially correlations within data. Whereas mechanistic models is what Jen was kind of talking about. Those models are built on the underlying biology of the system, and this tends to make them more robust. So you can apply these models to generate predictions in a wider variety of scenarios, whether that's environmental conditions or physiological conditions. They're easier to apply when situations change. Whereas empirical models like the NRC, they may not do as well when you try and put these equations in a different scenario or a different environment. They might not perform as well. So that's one of the advantages of a more mechanistic approach as opposed to something empirical like the NRC. And additionally, with a lot of mechanistic models, you can make those more dynamic as opposed to being static. So we can generate predictions over a longer period of time, so longitudinal predictions. So you can look and see how the things that you're trying to predict change over time, as opposed to just getting one static prediction. So it gives you a little bit more granularity in what you're trying to do. So acknowledging all of this, there was some work done by Tulutke in 2011, and what he did is he acknowledged the limitations of the NRC, which is really the main resource for horse owners trying to figure out what to feed their animal. It's one of the more well-known things out there. He acknowledged that and, knowing the advantages of using a more mechanistic approach, developed a model called FANCY, which is sort of a first stab at an equine mechanistic model. In this case, mainly to predict dietary energy or digestible energy and crude protein requirements of horses. So he built the base model structure in Excel and used NRC data and NRC equations to sort of generate this sort of first gen equine mechanistic model. The downside with this one, however, in terms of applying it, is it's not publicly available. So it's not incredibly easy for the average person to go ahead and use. And also in the paper, which is available, Telecki also acknowledged some of the challenges and limitations with this first generation of model, although it was a really great first step in the right direction to identifying kind of where we can go with mechanistic modeling for the equine sector. I should also acknowledge that these are not the only two models out there for horses. There's also a few other papers out there, but these particular ones are a bit more situation based. So for example, the paper at the top there, that's a mathematical model to predict digestible energy intake of brood mares based on body condition change. So it's a bit more of a specific scenario. Likewise, the paper below is a compartment model looking at how digesta retention works in the equine GI tract. So it is still technically a mathematical model for equines and should definitely be acknowledged, but it's very much situation based and not necessarily a whole animal model, which is really what we're going for here. And Jen touched on why we sort of want to advance modeling in the equine sector, and more specifically, what has kind of come about in a few studies and surveys. Jen mentioned the survey by Mad Barn, but there's also a few other papers out there as well that demonstrate that modern equine feeding practices often undersupply key micronutrients, but really importantly, oversupply macronutrients, in some cases a lot and really prevalently. So for example, energy and protein can often be oversupplied in equine diets, which can be a problem for a number of reasons. Oversupplying energy and protein can lead to obesity or overconditioned animals, and obesity in equines, so horses and ponies, for example, can be relatively prevalent. So prevalence has been estimated between 26 and 62% in some studies for obesity, which I personally think is a little bit high. And obesity is a problem in and of itself, but obesity is associated with other health conditions like, for example, laminitis, which leads to euthanasia and makes up a not insignificant proportion of equine mortality. Looking aside from the animal and looking at the environmental sustainability, overfeeding, and especially overfeeding things like protein, firstly is an unnecessary use of commodity crops that could be used for something else, and also leads to excess waste and nutrient excretion. For example, excess nitrogen excretion into the environment, which is an environmental sustainability issue. So that's all to say, there's a lot of reasons why we should maybe try and work to improve equine feeding practices and provide better, more accessible tools for people to come up with feed formulations for their animals. To that end, my work is really on developing the mechanistic model side, which, like Jen said, is one smaller piece of the much bigger puzzle that we're all working on here. So I'm just going to share sort of what's been going on the mechanistic modeling side of things and how that might play with the other pieces of this project. But really my objective was to develop a mechanistic model to describe the post-absorptive nutrient dynamics in the mature equine, which sounds like a mouthful, but really isn't that complicated. But as is probably clear to you now, there's not a lot out there for equine models to start from. So we can't really just take a established equine metabolism model from 2005 and build on the bones of that. We really need to look to other species and figure out what's out there, what works well, what's well documented, and what can we use to build the bones for our equine metabolism model. So what we decided to do is actually work on this model, which is actually describing calves. So this work builds on work done by Walter Garrett's, where they described, and colleagues, where they described a model integrating protein and energy metabolism in pre-ruminant calves. And this was further worked on by Dr. Chris Powell, who was a postdoc here a few years ago, as well as Dr. Jen Ellis and other collaborators, to create this mechanistic veal calf model called Simon. And Simon is a model that is coded in a program called SMART. And SMART is an interface program, so it's a point and click sort of program. But this is a really well established, well described model for metabolism, among other things. So we decided we should work on the bones of this and then adapt it for our equines later to give us a starting point. This is what Simon looks like in terms of a flow diagram. So this looks overwhelming, and I'm not going to go through all of these boxes and arrows here, but this is just to show you that Simon is big. It's a big model. It's got a lot of moving pieces to it. And we really had to look at this and figure out, what do we need? What do we not need? What doesn't apply? And how do we make this work for us? Because like I said, Simon was coded in a program called SMART. So it's an interface point and click program. But one of the first things that we wanted to do was actually make our model work in Python, because Python is very accessible. It's being highly uptaken in the modeling field as well as the other animal sciences. So first thing that I had to do, and this is a lot of what I did last summer, was take coding from Simon and SMART and translate that into Python before we could figure out what pieces we're keeping, what pieces we're leaving. And this took a little bit of time because an interface program like SMART, they kind of keep the code stuffed under the bed. So I had to go through and pull out the coding for what I really needed. But this model, I can walk you through sort of how this works. Really, it starts with an animal eating food. And in the case of a veal calf, this food is usually milk replacer, concentrate, and some forage. And then what's described here is how that food is digested by the animal and how those digested nutrients flow into the post-absorptive environment. So those post-absorptive metabolites like amino acids, glycogen, glucose, etc. And then how those metabolites contribute to changes in body composition of the animal. So the amount of muscle the animal has, the amount of fat, and all of those sorts of things. So right off the bat, structurally, we knew that there were some things that we have to change, because we realized calves are not the same as horses. So see, even I knew this coming from poultry. I knew calves are not the same as horses. So we know that there's some things that we have to change. The big one, and this is not because horses do not digest food, they do, but they digest their food in a different way than a veal calf, right? They don't have a rumen. And this is a big chunk of the model, is just that digestion part. And really our focus with my particular project was to develop the metabolism side of things. So instead of trying to work on two major modeling components at the same time, I'm sticking with the metabolism part, getting that working, and someone else is going to pick up the digestion modeling. And then ultimately we're going to stitch those two things together and have a really big comprehensive model that will look like this. But that's not where we're at yet. So we knew right off the bat that this digestion stuff, we're going to sort of just take it out and put it on the shelf for now until we can get to it. So that was one of the big structural things that we had to change. There's also some other structural things, like for example, the average adult horse is not drinking milk replacer. So we had to get rid of some of those fluxes as well. But really, a lot of the bones stay the same because metabolism works relatively in similar ways between species. So we end up somewhere like this, which is probably a lot easier to look at. This is a simplified version of where we're at with the equine metabolism model, but you'll notice a big piece missing, which is that gastrointestinal tract portion. So that's kind of been parsed out of the model, which results in us having to make some assumptions. So our animal still eats food. In this case, horses mainly forage-based diet with maybe some concentrate, no milk replacer, but we have to make some assumptions about how that food is digested and what happens to that food, which is fine. We're at the stage where we have to make some assumptions in order to get this metabolism part up and running. And then down the road, we can develop the digestion kinetic model and provide a lot more granularity to that particular part. But for now, we assume we know how these nutrients are digested, and then these flow into the digested nutrients part of the model. So your digested fat, your digested sugars, your digested protein, your digested energy. And then very similar to Simon, these digested nutrients flow into the post-absorptive compartment, so we can track post-absorptive things like fatty acids, glycogen, glucose, amino acids, and then how those metabolites contribute to overall body composition. So for example, body fat, different parts of body protein, so muscle protein, hide protein, visceral protein, etc. And then this body composition obviously contributes to the overall growth and development of that animal. How this works computationally, and I'm not going to slap some Python code up on a screen because I don't think anyone wants or needs to see that. But just to go through sort of how running this works, there's really three main parts to making this model work. There's the first part at the beginning. Oh, that's not going to do that. It's going to do that. Where we have to sort of set the stage for running the model. So we have to tell the model what we're starting with. We have to, there's a separate part of the Python code where we essentially put together the information that we know about the animal we want to simulate. So how old the animal is, its sex, its starting body weight, its body fat or its body condition score. And then we put that together with a feed bank. So we have a feed bank that's graciously provided by our industry partners. And that allows us to say, okay, this animal is going to be eating feed composed of these two ingredients in these two proportions. And then there's a little piece of code that basically generates all of our starting values to run our metabolism model. Running the model is in four main chunks, and I'll really quickly go through them. But really, it starts like a lot of any other statistical programs start with, again, setting the stage for the simulation you want to run. So we start the stage with how long do we want to run the simulation for? So for example, do I want to run the simulation from 0 to 35 days? In this part of the code, we also have all of our constants. So any constants that we need for any equations down the road. And this is also where that animal and diet information comes in from that part that we did earlier. And then we move on to the real engine of running the model, which really has the bulk of the model code. And this is where all of those equations and our constants end up to basically describe how those pools of things that we're trying to quantify change over the duration of the simulation. So it's a big, long list of equations describing all of those little arrows in those flow diagrams. And then it's sort of padded at the end by a couple of for loops. The first for loop is the solver. So this is the one that essentially steps through the model and provides us with the sizes of those pools at each interval in the model. And this allows us to have sort of a continuous output for those pool sizes over the duration of the simulation. And I'll show you graphs in a couple slides for what that looks like visually. But what this does is it basically just walks along between that 0 to 35 days and works on the equations and gives you the values for all of those pools for the duration of the simulation. And then the neat part that we added is an additional for loop that allows you to run multiple simulations. And this just means that I can run one animal, and then I can also run the next animal within the same run. And that's all part of the input code, but I won't get into too much detail about that, except say that this part was actually adapted from a dairy model, a different dairy model that was some work from a previous student in Jen's lab. So we have pieces leveraged from a lot of different species actually encapsulated in this code, in addition to that Simon model. And actually some work from a turkey model too, I think is stuck in there somewhere. So we have a lot of cross-species inspiration coming into this to make this actually as good as we can make it. And then this comes together once you run the model code. You can view the results, and this is the part everyone likes. It's a nice, easy part, so you can visualize your results in a CSV file, and you can look at them visually. Don't worry too much about the numbers on this. This is just to show you what the results could look like. So basically, you get your pool sizes spit out for the duration of your simulation, and you get this nice continuous reading of your pool sizes. So for example, for the duration of the simulation, under the conditions that I've given the model, so my particular animal fed this particular diet, this is how its live weight changes over the simulation. This is how its muscle protein changes over the simulation. And even the things like the metabolites. So this is how the amino acids or the glucose change through the simulation. So we can track a lot of things in quite a lot of detail, provided different animal, environmental, physiological scenarios. So for example, I could run this again with a completely different animal information or environmental information, and these results would look very different because I'm adapting it to a particular scenario. But this is a starting point. So we've tackled kind of the structural differences in adapting this model from Simon. But now we have to look more closer at the biology. And this is sort of the phase that I'm in now is computationally, our model works. It runs, it spits out results. It's lovely, it's beautiful. But we have to figure out if it biologically makes sense, which is really sort of the nitty gritty of building a model like this. Like, yes, I'm getting values, but are those values representative of a mature horse? Do they actually make sense? And if, or more likely when, they don't make sense, we have to figure out why. And this could be related to things because horses are not the same as veal calves. They're not the same as dairy cows. They're not the same as turkeys. So we have to figure out in those equations what might be different. So for example, one of the more obvious things that we're thinking of is veal calves are growing animals. So they're young, they're growing, they're fast. Mature horses, not so much. So we might need to look closer at some of those equations, especially those related to protein deposition or fat degradation, and figure out, okay, would those kinetics of those equations actually be the same in a mature horse? And there's also some other things that we need to look into. For example, we axed out the whole gastrointestinal tract. We have to see if there's any biological consequences of doing that, as well as species-specific differences. Equines are not necessarily the same as cows, and look into just really the biology of all of these equations, because computationally it runs, and that's half the battle. But now we need to make sure that's actually biologically accurate in order to be any way useful to anyone. And it could be something else that I haven't even mentioned. There could be something else in there that is not quite right. So really, it needs a very careful audit to make sure that we're doing something right. So yeah, these are sort of my ongoing next steps, is really digging into these equations and checking the biological validity of the model, and also evaluating with some literature data. So looking at the predictions, for example, of live weight or body fat, and comparing that with literature data and seeing, do these make sense? And like one of my old advisors loved to say, it's called research because you have to research. So that's what I'm going to be doing for the foreseeable future. And I'm going to leave you with this picture of this incredibly photogenic horse, which is actually from an article. I think it's 12 horses with better hair than you. So if you want to read that on your free time, but I'm now going to pass it over to Kara.
Okay, thank you, Emily. So we're now switching gears a little bit, and we are actually going to talk about some in vitro work that I have been working on. And so we'll go over a bit of background in vitro digestion, because there are multiple ways that you can approach modeling an in vivo digestion process in lab. So there's the static methods. These are pretty common methods that you'll see published in literature. And these include your ingredient or diet, as well as your acid, base, buffers, and enzymes being added to a set of beakers, digestion vessels, whatever you're working with in that case. Oftentimes, it may replicate digestion, which occurs in the mouth, then into the stomach and small intestine. And common methods include our pH consumption or pH drop methods. You might also know as pH stat. And then enzyme focused, so pepsin pancreatin, as well as two-step digestion processes. So these are actually two different processes, although they're quite similar. When we talk about a two-step digestion, it may not utilize the same enzymes as the pepsin pancreatin method. Whoop. But we also have what are called dynamic or artificial gut systems. So these are more recently developed, and these are a dynamic in vitro system. So unlike our static in vitro system, where once the enzymes and solutions are added to the beaker and they begin digesting, they're static, as in we're not adding any other solutions or enzymes. We're simply measuring the digestion or degradability at that point. However, in our artificial gut systems, what is really cool is whether it's vertical, horizontal, or a beaker system, the acid-base enzyme and peristalsis is controlled by a computer model. So we take published literature, published values such as changes in pH throughout the gastrointestinal tract of different animal species, enzyme activity, etc., and a computer model is developed. And this is what controls the addition of the acid, base, and enzymes, as well as the mixing of the samples throughout. So it's dynamic because the amount of acid, base, enzyme, etc., is actually being added throughout the digestion process. So whereas in our beakers, once the mouth portion has stopped, we cease enzymatic activity, add our next solutions in. In the artificial system, the computer model is telling it when to move on. So for example, in the stomach compartment, it might digest for an hour or two, but that flux is actually going through into the small intestine throughout those 2 hours as well. So we have gastric emptying throughout that time point. Then you also have the control of, say, your base as that gastric portion enters the small intestine. And there are pH probes that are also measuring pH. So this helps determine whether or not further base needs to be added or an acid needs to be added throughout the digestion period. So I'll go into a few more of our static methods first before we really delve into what my research is focusing on, because static methods are still the most common approach currently being used. pH drop is a relatively simple approach. It's a short run study. You can do it in a map. You can actually run a number of ingredients in a day. It maybe takes an hour or two, including prep. And what happens is we start with a pH of 8 in our solution. So this includes our ingredients as well as whether it's HCl, a water HCl combination, etc. And then an enzymatic solution is added in. And once the enzymatic solution is added in, you allow the system to digest for approximately 10 minutes. And at that 10-minute time point, for another 10 minutes, you measure the drop in pH over time. So that's why it's called pH drop. And once this digestion is finished, digestibility is calculated using regression equations to determine digestibility based off of this pH drop. So it's relatively inexpensive, and it's also a really quick method for evaluating digestibility. However, it hasn't been used across the board for all nutrients. Generally, this is widely utilized when investigating protein in vitro digestion. So it is something to consider if you're looking at the pH drop method to utilize. There's also the pH stat method, and this is quite similar to pH drop as well. It's a quick method. Again, you can run multiple ingredients in a day, but rather than measuring the change in pH over time as it drops due to digestion, you're actually measuring the maintenance of the pH. And so what happens is you're measuring that volume of solution, so your sodium hydroxide or your basic solution, whatever may be added in, to maintain that pH of 8, and you measure it from your 0 time point over to 10 minutes. Similarly to our pH drop method, you're going to use regression equations to calculate digestibility. Then there's a pepsin pancreatin digestion. So this is more involved. It's a longer digestion period, and there are more steps to it. But what's nice about the pepsin pancreatin digestion is it's trying to replicate what's happening in different digestive compartments. So the mouth, the stomach, or the small intestine. And this, for the purpose of this presentation, we're just focusing on the gastric and the small intestinal phase. But what happens is we start with a gastric phase and generally a solution of HCl and pepsin. And this is going to initiate digestion of the food and initiate protein digestion specifically. Then you move on to the intestinal phase. So what happens is you're going to cease enzymatic activity within the stomach compartment, and this is done by increasing the pH through sodium hydroxide is the most common. However, there are other basic solutions that can be utilized. Then a pancreatin solution, generally made in a buffer, is added to your beaker. So you don't remove anything from the gastric portion, rather you're adding the solutions in to what is remaining from the gastric digestion. And this is digested over a period of time, and you'll see why I say over a period of time rather than a specific time point, because that is what my pilot trial is trying to answer. Once that intestinal phase is done, you can cease enzymatic activity if you want to measure the proximate analysis of, say, the substrate, or if you're taking degradation measurements and you don't need the substrate, then you simply determine digestibility or your degradation through analyzing your samples and mathematical equations, which are used to calculate that digestibility or degradation. But there are some advantages and limitations with the static method. Same with the dynamic method, but because we're not utilizing that method, I won't get into too much detail for that. The advantages of our static methods are that there are no ethical concerns. So the great thing about this is once you're ready to run in lab, you can get going. You do not need animal care approval. There's no animals to care throughout the trial. If a run goes wrong, you can restart that run. So we're not worried about the ethical concerns or the use of animals. It's also quite reproducible. There are many studies which have investigated intra as well as inter-lab reproducibility with high correlations. And it's controllable. We can control every single aspect of that digestion, what temperature it's being digested at, what is the pH of our solutions at different times, how much enzyme is being added in. As mentioned, most of these are quite simple as well as rapid. So even though the pepsin pancreatin is still more time consuming than some of our other static methods, when we consider the time compared to running in vivo, we don't need to do diet adaptation. We don't need to sample over multiple days. We can take and gather all of our samples within 1 or 2 days. And it's cost effective. However, there are some limitations. For example, it's not representing the dynamic nature of digestion. So that's where those dynamic or artificial gut systems have an advantage over our static methods because they're trying to replicate that dynamic nature of digestion, that change that's occurring throughout those processes. There's also no measure of nutrient absorption. So we're not looking at what's disappearing from the GI tract because it's simply being released into the surrounding solution. There's no physiological feedback loops either. So we aren't looking at hormones, for et cetera, or hormones or other enzymes, et cetera, that are going to provide feedback to the animal and which may influence digestion and degradability. And finally, one of the big things about our in vitro methods is it's lacking microbiome. So when we look at fermentation studies, yes, you're using inocula that has a certain microbiome, but in our pre-cecal or our foregut digestions, we're not adding any microbiome. And there is discussion that there is some microbiome activity in the stomach of horses, for example, and that wouldn't be captured currently in the in vitro systems. So why? Why are we investigating this specifically? Well, there's minimal understanding of digestibility and fermentability of common feedstuff for horses. Now, there's definitely more fermentation data out there that's easier to measure, or total tract digestion data, but we don't cannulate horses the same way we cannulate pigs, for example, to measure intestinal digestibility. And there's less research compared to other animal species, specifically our agriculture production species. So for example, compared to cattle, swine, and poultry, which have quite a bit of data published, and there's been more in vitro digestion investigation into these species, there's very little on horses. I think right now I've summarized 6 papers total, and they're all over the place. None of them have any agreement. Some use one approach, another uses another approach. So there's no standardization across these methods like we see in other animals, especially actually humans. So I didn't mention humans before, but humans are really interesting because there's now an in vitro digestion approach called InfoGest, which took a number of scientists, enzymologists, et cetera, and they standardized an in vitro approach to analyze nutrient digestion for humans. What are these values going to give us? When we look at our digestibility and fermentability, this is going to provide the nutrients available for absorption from food. So this can feed directly into our digestion model. But degradation data is just as important because this is going to provide information on the rate of degradation of the ingredient. So how fast those nutrients are being released for absorption in the different digestive compartments. And this can help provide insight for feed efficiency as well as quality of feed. So not only are we generating coefficients to feed into a model, but we're generating coefficients to help with other aspects of nutrition as well. So applications for our equines. How are we going to apply this? Oh, Is it? Okay. No, yeah, oh, there we go. And now we just, oh no, I've been pressing the wrong arrows. That's why. There we go. So, importance of feed degradation. You'd think after how long of working online, et cetera, I'd know how to do stuff like this, but clearly I don't. Understanding feed degradability is a key factor in feeding our animals. So when we think about feed not only has its nutrient composition, but each feed ingredient has its own specific characteristics. This can be water holding capacity, specific characteristics on stability even, etc. And these are all influenced by multiple different factors, including crop management, geographic origin, harvest storage, soil nutrient composition, plant variety. And all of these factors can ultimately influence how feed degrades and how it interacts within the gastrointestinal tract. So different feed during digestion may have different nutrient interactions. For example, some feed may have fat that's trapped in a starch molecule or protein that's trapped in a starch molecule, whereas our fibrous forages, there might be interactions due to fiber within the gastrointestinal tract. And this means that each feed ingredient is going to have its own rate of degradability. By measuring feed degradability, we can develop inputs for models, but we can also use this for feed formulation. And I think the feed formulation stands out because, especially in the equine sector, unlike, say, swine production, where the entire herd is being fed the same diet, you can go to an equine barn, you can go to a horse barn, and each individual horse is going to be eating something different. Now, they might be on the same forages and pasture, but one owner might be feeding concentrates. Another might be feeding concentrates and electrolyte supplement. One might be feeding concentrates and top dressing with molasses. One horse might get soaked or steamed hay, while other horses are consuming dry hay. So we also need to consider that when we're talking about how do we feed a horse. The research objectives for my specific project can be broken down into a couple different goals. The first is our overarching goal, and that's to support the development of mechanistic models, specifically the digestion model, for the equine sector to advance the field of equine nutrition. But when we break my project down into the further research objectives, my specific project goals are to assess the current in vitro protocols in equine and compare them to other protocols used in other species. So we have drawn heavily from swine, ruminants, and poultry, as well as humans, to help develop our model and make it more robust. And then we need to select the most appropriate in vitro approach in relation to equine digestive physiology based on goal 1A. And that's where I'm currently at, is we're working on our pilot trial because we've selected what we think is the most appropriate approach, but we still have some questions that need to be answered. And then we're going to use these in vitro protocols to estimate nutrient digestion and degradability in the stomach and small intestine and measure hindgut fermentability of common feedstuff for horses, specifically North American feedstuff. And those results are going to be used as the input for the equine digestion model. So how am I planning on achieving these specific goals? Well, when we break down the research objectives, the first is the assessment, comparison, and selection of an in vitro protocol to simulate pre-cecal digestion. And then we're going to use that protocol to digest 32 feedstuffs and analyze for starch, protein, and fat degradation using protocol 1. We'll also be looking at digestibility by conducting proximate analysis as well as other nutrient analysis, such as sugars, on our final substrate that will be dried once we remove the liquid digested fraction. And then we're going to use the same approach for hindgut fermentation. So although I'm focusing on pre-cecal today, there is the fermentability aspect that we're also investigating. So we'll assess, compare, and select an in vitro protocol for hindgut fermentation. And again, we're going to ferment 32 feedstuff, and specifically our focus is going to be starch, protein, and NDF for our fermentation portion of the study. So now I'll get into a little bit more detail on what I've been working on in terms of the pilot. I think anyone who's seen me lately probably thinks I'm going a little bit crazy, and I am, and you'll see why in a second. So the 32 feedstuff of interest, we've split up into our grains, which is high sugar and starch, high protein ingredients, high fiber, and forages. And we wanted a variety of ingredients, again, because these can all have different interactions within the gastrointestinal tract, and they're going to degrade at different rates. So the proposed approach is relatively similar to what you would see in most static pepsin pancreatin in vitro approaches. We start with a finely ground substrate. Now, there's a lot of debate. We are getting some excellent help from Dr. Matt Nosworthy over in the Agri-Food Center. He has done a number of in vitro studies focusing on protein, but one of our discussions was, is it ideal to do finely ground, or do we do a particle size that would be more similar? We've decided to go with finely ground because that's what's currently used in most in vitro approaches. And we wanted to really focus on what our specific question was, which is degradation as well as digestibility. Then, once we've got that really established, you can go back and investigate. Let's look at different particle sizes. Does that change? So we start with our finely ground substrate. And this is going to start us off for our gastric digestion. So we add our HCl pepsin solution. We went through multiple different equine studies, or the multiple being the 6 equine studies, and we tried to see if there's any standardization across enzyme activity in the studies, and there wasn't. So we tried to match what's physiologically occurring in the horse to the best of our abilities for our pepsin activity. And this is mixed for 2 hours at 39 degrees Celsius. And this is a relatively common digestion time frame for many animal species for in vitro replication, but also in our equine studies. Where our big question came in is when we move to our intestinal digestion section, how long are we incubating and mixing our samples for? And this is because some studies used a 2-hour incubation, some studies went all the way to 18-hour incubation. And because we want to capture degradation of our ingredients, we need to make sure that that digestion time is sufficient to capture our degradation plateau. And so the objective of my specific pilot trial is to look at, do we need to digest our samples for 18 hours? Can we digest them for 12 hours or 8 hours? And what is the sampling frequency we need to implement so that we can capture the plateau on a graph? And this is where I've been going a little bit insane. So in my pilot trial, we do 20-hour digestions, which is really closer to 24 hours by the time you do all of your prep. And for sampling frequency, I sample at 0, 5, 10, 20, 30, 60, 90, 120, 150, 180 minutes. So it's pretty intense at the beginning. That's because that's where most of the degradation is occurring. But because we don't know where this plateau occurs for the various ingredients, and we assume forage is going to take longer than, say, our whey concentrate, which is readily digestible, or our cornstarch, which is or our corn, which provides a high source of starch rather than, say, fiber, we don't know what that time is. And after 180 minutes, I sample hourly until 18 hours. So there's a lot of data coming out of this, a lot of time points. But what we're going to do is once we plot this data, we want to compare our 18-hour, our 12-hour, and our 8-hour time points to figure out what is our digestion or an incubation time for intestinal digestion. Similarly to other studies, this is going to use a phosphate buffer and a phosphate pancreatin solution for digestion. And afterwards, we will wash our substrate so that we can dry it and freeze it for proximate analysis. And then the specific aliquot samples that are taken so that we can plot degradation are going to be analyzed for a couple different nutrients, specifically protein, starch, and lipid. Our protein analysis is going to either be done via OPA or nitrogen combustion. So OPA, of course, I'm blanking on the full name, but it's attaching, it's flagging the amine groups of our amino acids to analyze total protein. So within this pilot, we're also going to do a comparison of OPA and nitrogen combustion to see, can we use nitrogen combustion to adequately measure the amount of protein that is in our liquid aliquot sample, or do we need to do OPA? And this is because there may be other nitrogenous components that aren't feed related that we're capturing in combustion, but with OPA, it's only capturing that protein because it's tagging the amine group of those amino acids. However, OPA takes much longer because similar to amino acid analysis, you need to do a hydrolysis process, which is over a period of a day. Whereas with our nitrogen combustion, we can run all of our samples readily, whether they're fresh or they're thawed and frozen, or frozen and thawed, other way around. Starch analysis is going to be analyzed through glucose assay. This one was relatively easy to determine, and this is because there's been quite a bit of research on the human side as well as the animal side for starch degradation in vitro. And then lipid analysis will be through a fatty acid assay. And this is because we don't need the individual fractions, so GC is going to take a lot more time. We would like to get this data rolling pretty quickly. And so we just need our total fatty acids that have been released into the surrounding liquid. Similarly, in the intestinal digestion, the samples are going to be analyzed like our gastric digestion samples. And this is going to help us determine at which time points we need to sample. So most likely we'll need to do that rapid sampling at the beginning regardless. However, maybe we don't need our 150-minute time point, or we don't need our 240 time point. We can eliminate that. And that will help eliminate the number of samples total you need to take from this in vitro system. But because we don't have data on it yet, we're sampling every hour so that we can figure out those ideal sampling time points. And then for nutrient digestion, at minimum, we'll investigate dry matter, protein, starch, and lipids. However, the ideal is to investigate all of those, including ash and total sugars. So the next step specifically for this project is to analyze the pilot trial data. If any of you are going to ASAS, CSAS this year, I'll be presenting my pilot trial data there, so come check us out. I'm also going to be utilizing the data from this pilot trial to refine the in vitro pre-cecal analysis and digestion. And then we're going to digest a variety of feedstuff to determine the degradation kinetics and digestibility, and that was the list of 32 feedstuff you saw previously. So overall, it's a relatively large project in terms of what we're looking at to investigate. Most in vitro studies focus on maybe a couple ingredients, but this will generate some really interesting coefficients that can be used in different areas of the equine nutrition sector. And if you've got any questions, you can reach out to me, and I'm always happy to chat. Thank you.
So I think I only have like one more slide and then we're done, which is good. Hopefully that was interesting, gives you guys a bit of a glimpse about what we're working on. We of course can't do this alone, so we've got quite a few industry financial partners, as well as non-financial scientific contributions partners. So we definitely couldn't do this without all of this help from a variety of expertise areas. And that's it. Thanks. So I mentioned we have a couple of YouTube videos, thanks to Mad Barnes production collaboration. I don't know what to call it. So if you want some additional info on the project, they're there. I think there's going to be another one coming out maybe on Kara's stuff in the future as well too. So you can see those, I think, on the Mad Barn YouTube channel. And that's it. Thanks, guys. So we have a few minutes if anybody has any burning questions online or in person. Yeah. Like that. Okay, question for Emily from Katie online. Okay, the equine industry is perhaps one of the few where there's a tremendous diversity in mature size, so 10 plus fold difference from minis to heavy draft, and likely needs to function with a much larger variation versus other livestock. Have you looked at this variation yet, and how good are these models dealing with this variation?
Great question, Katie. In summary, haven't looked at it yet. Right now, we're trying to get the model working on just your average run-of-the-mill maintenance horse. But there are equations in the model that actually incorporate the percent of mature weight that the animal is at, which informs certain deposition rates and things like that. So it is built in there, but I have not played with it too much yet. But you are exactly right. I have learned that, yeah, horses vary a lot in a lot of different things, which they're not as nice and uniform as I'd like them to be. But yeah, no, that's a really great question. Thanks, Katie. Shannon is very proud. Yeah, Anna.
So follow-up question for you.
Okay.
So you say a lot the word like general horse population or average horse.
Yeah.
Population. And I totally get where you're coming from, but especially in Canada, our average horse population isn't so average. Kind of following up on Katie's comment that there's a lot of variation, especially looking at breed variation. We do know that there actually is just metabolic differences between different breeds.
Absolutely.
And then diseases on top of that. So you mentioned like EMS quickly. So how are all of these kind of exterior factors that have really big implications on the individual horses being taken into account?
Stay tuned. They are not yet. They're not yet because we're trying to get it working on a normal, I know, maybe average general is not the best word to use, but a particular type of horse. We want working on a particular type of horse before we start delving into differences between breeds or differences between disease states, or even exercise is also another really important thing to incorporate into the model. But it is well established, maybe not so in equines, but in other species, how to model those sorts of things in a metabolism sense. So how to incorporate a disease challenge, for example, into a model, or even just toggling between breeds. That's I'm not going to say the word easy, but that is relatively common to incorporate in these sort of models. And that does adjust coefficients of certain equations. So certain breeds might deposit muscle faster than others, and some breeds may be fatter than others, for example, or like Katie was saying with the mature weight. So there are ways to toggle the equations within the model based on certain breed situations, health challenges, even environmental conditions is also another big one. Like, for example, a heat challenge or a cold challenge, and how that affects basically how the animal grows or maintains weight. So it's to come, but it is not there yet. But great question.
I'll add to it. We'll make those changes as the model fails, because all models are a simplified representation of reality, right? So it's like, we have our base model horse that the model's going to be built for. And then, okay, let's try it with a small pony or a draft breed. Does changing the starting weight of the simulation and the average intake of that animal and the average body composition of that specific breed, did those changes alone allow us to predict the right outcome for that animal? You try it in the model and you find out. When the answer is no, then we go, okay, well, what's different physiologically about that breed compared to what we're simulating currently? And that's a great way of how models can also collaborate with experiments to say okay, what do we actually need to look at experimentally? How are these animals actually different? And so I think it's all models fail and some are useful, and we will build the base science and then you just keep challenging it with different scenarios, and as it fails and as there's necessity to improve it, then you advance it. So certainly in the poultry models that are out there, there can be specific sets of genetic parameters that are specific to different genetic lines. And so, like Emily said, you can kind of say okay, this is the genetic line that we have or the genetic type, and it's going to have a specific set of It could be maximal rates of protein deposition or, things like this. But then you can kind of like, advance it and simulate different animal types. But you start with the basics and then we build complexity. It's definitely, you have to start with sound science at the bottom, and then you can look at what alternate scenarios do you kind of need to be able to simulate. And certainly horses are a very eclectic collection of animals. So, building genetic differences in probably will be part of the future, but for the first iteration, it's can we get the biology correct and the metabolism pathways correct? And then you can look at beyond that, how do these things vary between different scenarios?
A lot of making it and breaking it again.
Yeah.
So what's your like base horse spec?
Right now, it's just a generic 500 kilogram maintenance.
Okay.
Like that's as far as we are right now. That like there's no particular breed assigned to that. It's a particular maintenance horse starting at 500 kilograms with a certain percentage of body fat, but there's no particular breed assigned to that. And we're just assuming maintenance, so no crazy exercise or anything like that. And we're just assuming a typical maintenance diet I think that's from the Matt Barn website, the diet that we're using. But we will expand. Like I said, we will go into the literature data and take, for example, a different kind of horse and what they fed that horse and try our model, put those conditions into our model and see what our outputs look like. So we might get it working with my generic made up horse, and then I can test it with, some Italian heavy draft horse or something and what they feed those and see what our outputs look like. And it probably will be a little bit bonkers, and then we'll have to go in and adjust and figure out what's going on there.
But then the nice thing about, building this as a mechanistic model, yeah, we can do that one next. The nice thing about being a mechanistic model, because, you run into more of these like breed difference problems when you build an empirical model, because that's purely just based on correlations in a specific data set. And any model that you build off of that will predict really well on that type of data, but not necessarily well on a new type of data, right? So that's really, it's a problem for this type of model, but it's very much an empirical model problem. The benefit of going with this mechanistic approach is it's really based on underlying, physiological and metabolic pathways. And so, you may get more robust answers out of it with different scenarios. Like if the differences are just that we fed a different diet to horse type A and horse type B, then that may get captured in, being able to feed the model a different diet. It may not. It may be that there's physiological differences there too that we have to simulate, but it gives you a platform, a modeling platform to investigate what are the differences between these horses. Is it just diet? Is it just starting weight, or is it something else? So it definitely gives you a platform to explore those questions. Okay. Okay, for Kara, wondering how you're setting the best incubation times, for example, protein. How are you validating this? We have found enzyme concentrations are much too aggressive in vitro systems, etc.
Okay, thank you, Dr. Moore-Collier. So right now, we're using our pilot to help us determine. However, as part of this, we, when we were developing this system, we realized that there's a lot of questions we want to answer. And based off of actually the presentations at the EHNCC, we took some of those discussions into account when developing this pilot. That being said, for example, the best incubation times, we're really looking at that degradation plateau that we're hoping to find and meet. And we're also taking into account currently the information that has been published. Now, that being said, we obviously know that it's not going to specifically answer all the questions. Taking into account the discussion that was in the in vitro section in Belgium, we did go with a lower enzyme rate or a lower enzyme concentration compared to what some other studies have done. But again, this is what we're hoping our pilot trial will help us answer, and hopefully we can coordinate with some other researchers once we get this pilot data to go, is this an ideal incubation time point? And is this an ideal incubation, or should we go shorter? Should we go longer? Should we reduce the enzyme activity? Unfortunately, we can't answer all the questions we're hoping to answer within this specific study, but by coordinating with other researchers, we're hoping that we can take into account maybe what they have found and adjust the actual study down the line from the pilot study for that as well. So it's a pretty vague answer. I wish I had more specifics on how we're specifically looking or how we're adjusting incubations for that. But again, because we aren't looking at, well, we want to characterize these 32 feedstuff, one of the questions was, do we focus on looking at different enzyme concentrations that we're adding in the solution? Do we focus on the different incubation time points? And right now our focus is on the incubation time point, and the specific enzyme concentrations that we're adding in we may need to address down the road. So hopefully we can loop in with more researchers to discuss this. If anyone out there knows more in vitro researchers, we're always looking to discuss with people. But ultimately, right now, part of answering whether or not that specific incubation time is sufficient is looking at that rate of degradation, because we've measured it both for the gastric as well as the small intestinal sections. So we can even look at those different sections separately and go, okay, maybe small intestinal is sufficient, but something's going on with our gastric section. Maybe we need to reduce that level of enzyme that's going in, or we need to reduce or increase our incubation time point.
Maybe the only thing I'll add is just for the in vitro characterization of feedstuffs here, we're not trying to use it to predict digestibility of feedstuffs. We're actually using the in vitro simulation to simulate or to define the degradability parameters. So we're looking at the maximum potential of the feedstuff. Where we calculate digestibility is when you put those qualities of the feedstuff into the model, and those, for example, you might get the undegradable fraction, the soluble fraction, and the potentially degradable fraction, right? So that's what's coming in, that's characteristic of the feedstuff. Then we're going to have the degradation rate of that middle degradable fraction. So these are maximal potential values of the feedstuff that we're trying to get in vitro. When you put that potential degradability profile of a feedstuff into the model, degradation rate works against, for example, passage rate. The net result of that is to predict digestibility as the outcome of the model. So the idea here is that this is characterizing maximal potential of the feedstuff. And in the model, that's where we can look at things like, okay, well, what's the average passage rate? How is that affected by things like feed intake or, bulk density of the diet and things like that? So that's where we can bring in some of those feed attributes, into the discussion to net predict digestibility. So just to point out that digestibility is not so that's why we're not stopping our in vitro digestions at a specific early time point, which is trying to estimate gastric retention time. We want to run it till it gets to the plateau so we know exactly what is the undegradable fraction of that feedstuff, and then get the rates from the front end of the curve, if that makes sense. That helps. Good. Do you have a question too?
Yeah. There's another online question. And then
Okay.
Yeah,
sure.
You want to do one online?
Yeah, so the online was, do you plan on using anything similar to the TIM, modified to horse GIT, or more complex digestion approaches? So right now we aren't planning on using the TIM or the dynamic. There was a discussion about that. So what I didn't outline in this presentation due to time is one of the downfalls with TIM is it's much more complex. There's not many systems in Canada. Luckily, we have one at Food Science here, but there are a lot more hiccups that come along with the TIM. For example, I have had the TIM literally spew out probes where I've lost an entire run because now I have intestinal solution spewing out of this pH probe. It needs well-trained staff. So it's relatively easy to train someone on a two-step or pH drop approach. However, when we're looking at the TIM system, you need someone who's inherently comfortable and familiar with the system because of all of the different probes that are being utilized, the different pumps that are being utilized. If something breaks, you often need a technician to come in. So that automatically increases time. It also increases expense. So right now, we also want to have something that could be replicated across different labs with different equipment. So a shaking water bath is pretty standard across labs, but a TIM system is not. And so we want something that others can also replicate and then potentially modify down the line.
Last question?
Okay.
Because I know we've got right over.
I think I need just a comment about fermentation or incubation for the intensive or large intensive incubation. Food or feeds, he reached to large intensive after between 3 hours and 3 hours and a half to second to fermented mice. For example, if you don't talk about cellulosic bacteria for fiber, you tell maybe make evaluation for feeds about 32 feed stuff. Okay.
Yeah.
I have another question for this point, especially for incubation. Maybe the feed stay about 35 or 40 or 50 hours, not incubation. This is in vivo in the horse.
Yeah.
So I think you need to cover this area. All of this time should be increase your incubation in the in vitro trial.
For the fermentation?
Yeah.
Oh, yeah. So we plan on at least doing 48-hour fermentation, if not longer, for the fermentation study.
Point. For microbiome, you tell us you don't make a microbiome because it make in vitro trial, but you can take samples like a rustic ruminant. You can make this. You can make design for rustic technique, the same rustic technique in the in vivo in vitro trial, and you can make select the kind of bacteria like cellulitic bacteria, and you can make evaluation for this in vitro also.
Yes. So what I was talking about specifically with the microbiome is for the pre-cecal digestion, we tend not to add anything in for the microbiome, but there is in vivo data supporting that there might be some microbiome activity in the stomach. And could this influence? But for the fermentability, definitely we will be isolating microbiome for that.
Last comment. What you can do with fermented feed now in market, especially I am from in Middle East, in Egypt. Also, I have Arabian horses. Your friend talk about the type of breeds in Canada for pony, for large, for races, for rugby, like Dr. Ellis talk. For Arabian horses, this is the huge market in Middle East and the huge number, especially in our area. If you make a modeling or model for metabolism, we needed this, okay?
Mm-hmm.
But in especially for type of feeds or type of fiber, we have a big problem. If you reach for some result for this point, you cover a big problem for us, for almost the Middle East, Emirates, Qatar, Saudi Arabia. The most 3 countries have special horses. I think now the market for feed reached to $8.5 billion. If you make like this, I think you need more overseas support, plus your support in your country here.
Definitely. So, and this is where we can continue to work on this project. As the start, we're working with North American Feedstuff because Mad Barn is a North American company, primarily working with North American clients, so Canada and the United States. That being said, the hope is eventually, my hope would be to develop in vitro systems similar to the InfoGest in humans, where we have a whole bunch of researchers come together and collaborate and develop one specific methodology that can be utilized, that's very focused and is capturing to the best of the abilities the physiological parameters, because right now different studies are using a whole bunch of different parameters, but eventually it would be nice to expand out of North America and that's where we can start looking at other feed stuff down the line, and why we wanted to also make this relatively reproducible, hence a static method rather than a dynamic TIM method, because those can easily be applied overseas just as they would be applied here.
Okay. Last one, for fermented feed stuff.
Yep.
You put it in your blend to evaluate this feed stuff. Fermented, for example.
Yeah, so pre-fermented like a silage, right?
Not only silage. We have another problem, and especially now I have a student in Chinese academy. Almost China now work in fermented rice bran. Something don't eat it for some animals, but after fermented, something hardened is a byproduct from for wheat, for rice. You can using and make fermented inside fermented and using like supplemented for animal. This is new trend now, but before we using a little bit. Now, after problem in shortage after Ukraine and Russian war, we have a shortage in food, in feeds. So now we have another replacement for fermented feed stuff. We have a lot of type of fermented feed stuff. I hope if you suggested put in your blend to put it as a regulation feed stuff.
Yeah, so I think that, again, I think because we've got a lot of ingredients to analyze right now for the original trial, I don't know if we'd be looking at fermented feed stuff, but that being said, it can easily be applied to fermented feed stuff as long as we have access to those ingredients. And again, that's our hope, is that we can develop a method where, okay, someone wants to investigate fermented feed stuff. Well, now that the method is developed, it's not going to take very long to run one day of one ingredient. It's just the 32 feed stuff we picked are specifically more common ingredients that might be used here, either as concentrates, top dressing within a concentrate pellet, or as our forage sources.
Good. Awesome questions, guys. Okay, I know we went way over, so we'll leave it there. But thank you guys very much for coming in person, online. If you still have questions, feel free to reach out to myself, Emily, or Kara. And we can get back to you. I already sent out the invite, if you haven't seen it already, for the June and July CNM seminars, because I wanted to make sure it got in people's calendars before the summer got too busy. So I think at the end of June, we have Dr. Elijah Chiari's lab presenting, and in the middle of July, we have Dr. Trevor DeVries' lab presenting. So if you haven't got those invites and you would like to be on our mailing list, just reach out to me and I will add you guys. So great. Thank you guys very much. Have a great day. Sorry we went over. Thank you, everybody online.
Write that point down, because I always